stragglers. Okay, well, thanks very much. It's my pleasure to welcome Pieter Rein. I'll let you take it away.
All right, thanks, Jordan. Thanks for the, and thanks also to the other organizers for organizing this wonderful meeting. It's nice to be here finally after, what is it, 2 years or so? 2 or 3 years, yes. So, as you probably know, right, so cells, living cells, they live in a highly dynamic environment to which they continually have to respond and adapt. Right? And to this end, they have developed a range of strategies. One is the bed hedging strategy of maintaining phenotypic diversity at the level of the population. The other strategy is to detect the change in the environment and respond appropriately. But perhaps the most interesting strategy is to predict and anticipate the changes in the environment. Now, it has long been believed that the capacity to predict future changes in the environment, that is a property of higher organisms, but in the past 10 years or so, experiments have vividly demonstrated that even single-cell organisms can predict changes in their environments. And to just give you one example, experiments have shown that the bacterium E. coli can use the arrival of one sugar, lactose, to predict that the next one will arrive, maltose, as you can see here, and that moreover, this capacity to predict the arrival of the next sugar can provide a fitness benefit by increasing the growth rate. Right? So clearly, even single-cell organisms can predict changes in their environment. Now, of course, this capacity is only useful if cells can reliably predict these changes on the time scale to mount a response. Right? And so this raises the overarching question of this talk, which is how reliably can cells predict the future? Now, before I continue, I would like to thank the people who did the work. My PhD student, Agit Chalma, who's sitting there in the room. Postdoc Farah Galstian, former students Jeroen Goedhart and Lotte Slim, and our collaborator, Niels Becker. Now, the setup of the problem, right? So here you see an input signal in red, right? And the question of today is how accurately can the cell produce from its output, shown here in blue, how reliably can the system from the current output here predict this future input signal, right? Now, while the system needs to predict this future input, it can only sense the current signal and remember the past signal, right? And this immediately implies that the accuracy by which they can predict the future, the amount of information they can possibly have about the future, the so-called predictive information, that this is fundamentally limited by how much information they have extracted from the past signal. Now, what is important is that this information bound does not depend on the design of the system, right? This information bound is only a property of the signal, right? It's determined by the temporal structure, the temporal statistics of the input signal. However, how close the system can come to this bound, that does depend upon the design of the system, right? And so today we will be considering signaling networks of different topologies, right? And in particular, how close the cell can come to this information bound, that does not only depend upon the statistics of the signal, but it also depends upon the topology of the signaling network, right? Moreover, how much information the system extracts from the past signal that is determined by the resources devoted to building and running the network, right? So receptor molecules and reader molecules need to be synthesized on a timescale set by the growth rate. Moreover, these signaling systems are typically driven out of equilibrium, which means that also energy needs to be spent to operate the system, right? And these resources, right, this resource cost puts a fundamental upper limit on the information, the past information that the system can extract from the past signal, which in turn puts an upper bound on the information the system can possibly have about the future, the predictive information. So the questions that we will then discuss today are, what is the information bound, this information bound, how can we compute it? What is the design of the optimal system that can actually reach this information bound? How does this depend on the statistics of the input signal? How close can biochemical networks actually come to this information bound? And last but not least, what is the optimal strategy for extracting past information to maximize predictive power? Now, we will, for today, address this question by studying linear systems, right? And for any linear system, the output of the system X can be written as this integral over the input as shown here in red via this response function chi and this noise term psi, right? And what is important is that this response function that determines how the past input is mapped onto the current output, right? And this response function depends on the topology of the network, right? So this response function, that will determine what the signal characteristics are that the cell is going to use to predict the future. Now for today, we start by studying Markovian signals, Ornstein-Uhlenbeck, characterized by this two-point correlation function. So we have an input signal with a variance sigma squared s, which decays exponentially on the time scale tau l, the input time scale. We then obtain the information bound by maximizing the mutual information between the current output x and the future input s. That's the predictive information subject to the constraint of the mutual information between the current output x and the past signal trajectory s past, which is the past information, right? And then by maximizing this predictive information subject to the constraint of this past information, we can then obtain this information bound. Moreover, we also obtain the kernel of the optimal system that makes it possible to reach this information bound. And for this signal, the optimal kernel is given by a delta function, shown here, right? And what this does is that it maps the current input directly onto the current output. So this system with this delta function as a response function, it makes an instantaneous copy of the current input. It instantly copies the current input into the output, right? And that this is optimal can be understood intuitively because here we have a Markovian signal and all the information on the future signal is contained in the current signal. There's no information there's no additional information in the past signal that is not already contained in the current input signal. Now, from previous work, we hypothesized that push-pull networks should be able to make such instantaneous copies, right? And a push-pull network is shown over here. So we have a receptor that binds a ligand, and then this ligand-bind receptor drives a push-pull network, a phosphorylation-dephosphorylation cycle, for example, right? And so then what we decided to do is to map out these information curves for this push-pull network. And the first question that we then started to address is, does maximizing predictive power, does that imply maximally extracting past information, right? Because after all, the cell bases its prediction on the past signal. To make a prediction, the cell has to extract information from the past signal, right? So the first question you may ask, does indeed maximizing predictive power, does that imply also maximally extracting past information? Right? And we started by addressing this question by ignoring this operating cost. I will come back to that later. Right? So for now, the resource cost is simply the number of receptor molecules and the number of readout molecules multiplied by the growth rate. Right? And what we then do is then we maximize this past information for a given resource cost by optimizing this ratio of the number of readout molecules over the number of receptor molecules and this integration time tau r, which is basically the time it takes to go through this cycle of modification and demodification, right? And so when we then do this, right, we find this blue curve here, which gives the maximal past information as a function of this resource cost for this particular system, right? And then we did the following, which is perhaps not entirely trivial. We then asked, how well does this system then predict the future? What is then the future information, this predictive information for this system? And that's this blue curve shown up here, right? And yes? So here it's essentially the number, it's a copy number, right? The number of receptors plus readout molecules multiplied by the growth rate, which would be per unit time. Right. And so what we have done here, right? So we maximize the past information, the resource constraint, that gives this curve, and then we just ask, how well do these systems predict the future? What is their predictive information? That's this blue curve up here, and what you see that the system is quite below the information bound, right? So this immediately implies that optimally predicting the future does not imply maximizing the information extracted from the past. Moreover, it means that not all bits of information are equally predictive. Because if all bits of past information were equally predictive, then the system would be at the information bound. So then we decided to directly maximize not the past information, but directly maximize the predictive information under a resource constraint. And when we do that, we get the lines shown here in blue. And what you can now see is that the system is much closer to the information bound now, but still it's not fully at the information bound, right? So the optimal system that maximizes the predictive information under a resource constraint is not at the information bound, right? And again, this information bound is this black curve that maximizes the predictive information for a given amount of past information. Moreover, this also means that not all bits of past information are equally costly, because if they were equally costly, then these two bounds, the red and the black line, would overlap. Right? So combining the information from these two slides together, we can conclude that not all bits of past information are equally predictive nor equally costly. The question, of course, is now, why is the system not at the information bound? Right? And so what we then decided to do is to map out the region in this information plane that is accessible under a given resource cost. Right, so this green line here is the accessible region subject to a resource cost less or smaller, equal or smaller than 10 to the 4. Right? And again, this red line corresponds to the red line shown before, so that corresponds to the system that maximizes the predictive information under a resource constraint. The blue line corresponds to the blue line of the previous slides, it's corresponding to the system that maximizes the past information under a resource constraint. And what this figure shows, right, the 2 points to take away from this figure is that, first of all, the system can be at the information bound, right? And secondly, the system can increase both the predictive and its past information by moving away from the bound, right? And this is interesting, right? So why is this now? Well, to elucidate, we decided to analyze the design of the system along this green information front, right? And the result is shown over here, right? So when the system is at the information bound, this theta is 0, right? And what you can then see is that this integration time, tau r, is 0, and the number of readout molecules, xt, equals the number of receptor molecules. So here, at this information bound, the integration time is 0, and the system is an instantaneous responder, right? So at this information bound, the system copies the current input into the current output, and this is indeed what maximizes the predictive information for a given amount of past information, right? But the system can do better by increasing its integration time and by increasing this ratio, which means that given that the sum is essentially constant, right, it means that it trades receptor molecules for readout molecules, right? And this is really the reason why this predictive and past information can increase. Because, right, a readout molecule is either phosphorylated or unphosphorylated, right? So a single readout molecule provides one concentration measurement. It constitutes one physical bit that provides at most one bit of information on the concentration, right? And as you go along this information front, theta increases, right? You trade receptors for readout molecules, which means that you increase the number of concentration measurements, and really what happens is that you use your receptors more effectively, more efficiently, because really what happens is that you take more concentration measurements per receptor, right? And that is illustrated here. So here the system is at the information bound. If you then move away from it, right, you exchange, let's say, a receptor for a readout molecule, and now you have more concentration measurements in total because you have more readout molecules, right? So this effectively allows the system to take more concentration measurements for the same cost. It effectively makes the physical bits of information cheaper. But there is a price to pay, and that is that this integration time must increase, right? To keep these concentration measurements independent, the sampling interval must be longer than the receptor correlation time, because otherwise you would start taking redundant measurements, right? So to keep the measurements independent, the integration time, the cycling time, must increase. And that means that these concentration measurements, these samples, if you wish, they become older and hence less predictive about the future concentration. So there's a trade-off, right? So as you go along this front, right, you see that the number of physical bits of information per resource cost, that increases, and that is really because you use your memory, your receptors more effectively. But at the same time, the predictive information per physical bit decreases, and this interplay, this trade-off gives rise to an optimum that maximizes the predictive information per resource cost, right? So the optimal system faces a trade-off between bits that are most predictive and those that are cheapest. Now, so far, we've ignored the power cost. Now we're going to include the cost to run this phosphorylation, dephosphorylation cycle. And as you can see, this cost is really given by the free energy drop over the cycle, the delta G of ATP hydrolysis, over this integration time. And what we've already seen is that at the information bound, the integration time must be 0, and that's precisely where then this chemical power will diverge, right? And what you will see indeed is that when we include this operating cost, the optimum moves away, the optimal system moves away from the information bound, right? So what we've seen is the operating cost diverges at the information bound, and by including it, the optimal system, denoted here in red, moves away from the information bound. Now, in the last 5 minutes, I would like to address non-Markovian signals, right? So up to this point, we've discussed Markovian signals. Now we're going to look at non-Markovian signals. And the motivation is that organisms that navigate their environment with directional persistence, like, for example, E. coli, right, that they will experience non-Markovian signals with memory as generated by their own motion. Right? And what is interesting is that the response functions of these systems are typically not simple exponentials, but they are of the type shown over here. They're what's called an adaptive kernel or a derivative-taking kernel. Right? This system subtracts the most recent, from the most recent signal, the signal further back into the past. So this is really a derivative-taking system. Right? And the question, of course, is, can we understand the shape of this integration kernel? Now, to address this question, we then looked at the simplest class of non-Markovian signals, which is just a particle, an underdamped particle in a harmonic well, right? So L is the concentration, its time derivative is the velocity or the change in the concentration, right? And then what we assume, what we imagine, is that organisms that need to climb gradients, that they want to know the change in the future concentration V, the future change V, rather than the value, right? So this is not an outcome of a calculation, this is what we assume. It's the premise of the calculation, right? And so what we then do is we're going to optimize the system to optimize the prediction of the future derivative. And what we then find is that the optimal system to optimally predict the future change in the signal, that is actually a system that combines the current derivative, this second term, with the current value, right? And this is interesting, right? Because you would think that it maybe only consists of the second term, but of course, in general, also the current value can provide information about the future derivative. So in general, the optimal system combines the current value with the current derivative to predict the future change, and the degree to which it uses the value and the derivative, that's determined by the cross correlation between the current signal value L and the future change phi of tau versus the current change and the future change, right? These cross correlations, they determine the degree to which the system uses the value versus the derivative to predict the future. And now comes the point, right? So it's known from experiments, that E. coli can measure concentrations over a very wide range of background concentrations, over orders of magnitude, which is much larger than the typical concentration range over a single run, right? And what you can then show analytically is that then the cross correlation between the current value and the future change, that goes to zero, right? So this A goes to zero, and then the optimal kernel becomes a fully adaptive kernel that only takes a derivative, right? So we argue that E. coli wants to predict the future derivative, and because it can sense concentration over a very wide range of background concentration, the current value is no longer correlated with the future change, only the current derivative is still correlated, and for this reason, we argue E. coli has an adaptive kernel. So then lastly, right, so we then use this as our input signal, as measured by Mattingly and co-workers in the lab of Cherry and Monet, right? We looked at the E. coli chemotaxis network, which consists of this push-pull network plus this adaptive network implemented by receptor methylation on the timescale tau m, right? The optimization parameter now is the methylation time, so we keep tau r constant, and the result is then shown over here. So again, in black we have the information bound. It gives the maximum predictive information for a given amount of past information. The red line is again corresponding to the optimal system that maximizes the predictive information for this resource cost. And again, what you see is that the optimal system is very far away from the information bound, right? And to understand that, we look again at the design of the system along this front, and what you can see is that as you go along this green information front, the methylation time increases, so there's an optimal methylation time that maximizes this predictive information. Moreover, this optimal methylation time goes down as this resource constraint is relaxed and as the system moves towards this information bound, right? And this we can also understand intuitively, right? So all the information on the future derivative is contained in the current derivative. It's Markovian in the space of the derivative, right? But if the system would take an instantaneous derivative, it would also instantly adapt, right? And then basically the response goes to zero, right? Because if the system instantly adapts, the response magnitude goes to zero, the gain goes to zero, and then the response is swarfed by noise, right? So the system needs a finite methylation time to increase the gain, right? So the trade-off is between that on the one hand the system wants to take the most recent derivative because that's most predictive about the future, but on the other hand it also wants to take a derivative that is reliable, and for that it needs a larger gain and hence a larger methylation time. The last slide is then one where we now keep the resource cost fixed, but we vary the steepness of the gradient, so we consider cells that swim in an exponential gradient. The red line is, as before, the optimal system where we optimize over the methylation time. And the blue curve corresponds to the E. coli system where we just keep the parameters fixed as set by, as taken from the literature. And what you see is that when the gradient is large, right, that then the maximal predictive information, the predictive information of the E. coli system is significantly lower than the predictive information of the optimal system. And more strikingly, the past information is much larger than that of the information of the optimal system, right? And so this basically means that the E. coli system uses a lot of past information not very effectively. And this is in stark contrast to the situation at shallow gradients, right? Then the predictive information of E. coli is very close to that of the optimal system. It's very close. Moreover, it uses its past information much more effectively for prediction, right? So the chemotaxis system, we believe, is strongly optimized to predict future derivatives in shallow gradients. And this also makes sense because the shallow gradients are the hardest to sense, to measure, and to predict. Okay, so this then wraps up my talk, right? So biochemical networks can extract the most informative signal characteristics and reach the information bound, right? So these push-pull networks, they can copy the current input into the current output. This chemotaxis system can, in principle, take an instantaneous derivative. However, reaching the information bound is exceedingly costly, right? So we've seen that the power diverges and also the gain goes to zero. That's what we saw in the context of the chemotaxis system. And in general, it is not optimal to be at the information bound. Cells can increase their predictive power per resource cost by moving away from it, right? Increasing the integration time reduces the chemical power, enables more measurements, and increasing the methylation time increases the gain. But as this resource constraint is relaxed, as these resources become cheaper, cells can move towards the information bound. So this ends my talk, but I would like to show one slide, which is that we have an opening at our institute AMOLF for a new tenure tracker in theory. So AMOLF is a lean and mean science machine in Amsterdam, working on information in matter, autonomous systems, and energy harvesting materials. And we're specifically looking for someone interested in information, the topic of this workshop, learning, adaptation, and the machine learning aided understanding and design, and evolutionary computing and inverse design. So if you want to know more, there's a link to our website. And with that, I would like to thank you for your attention.
All right, so we'll open it up to young people.
Thanks for the talk. So you said at some point that the cell takes into account the first derivative of concentration in computing the maximum predictive information, if I don't make a mistake. So,
yeah.
so do you know if it takes into account higher derivatives, second or third? Just I'm wondering about whether or not it's optimized to the broader shape of the concentration gradient rather than just local information about, first
Yeah, so that's an interesting question. So I think, this system, right, really takes this this first derivative, it subtracts from the recent signal, this more distant signals further back into the past, right? But it does it really via this kernel, right, which has been measured in the lab of Howard Berg, right? So this is not, of course, a very simple standard derivative as you would normally take it, right? It's via this convolution. But I think if you really want to look at second derivatives curvatures, I think you would probably need a different topology. I'm not aware of signaling networks that can do this, but it would be an interesting question, and it would perhaps make it possible to determine whether it's reaching a maximum, for example, or not. Right? As the cell is moving towards the maximum, whether it can then at any moment sort of measure what the change in the slope is. That will be the second derivative.
And how steep are the typical concentration gradients in E. coli, so you said?
Well, so that we really do not know. Eh? So E. coli lives in a very broad range of environments in our gut, but also in the soil, in water, and so what the typical concentration gradients are is very hard to say. But what our work, I think, does clearly show is that this system is really optimized for sensing very shallow gradients, which I think makes sense because these are really the hardest to detect.
Hi. Yeah, thanks for the talk, sir. Sir, I wanted to understand what's the meaning of resource constraint and how is it controlling the optimization of the predictive information bound?
Yes. So, so what you see also with the push-pull network, but also here, right, is that so this is when this resource constraint is very tight, right? So this is the scenario where the system is only allowed to have 100 receptors and readout molecules in total. This is the bound when this sum is maximally 10 to the 4th. So this is sort of a typical number for E. coli, right? So this corresponds to the number of receptor proteins and readout molecules as found experimentally. And this is what it would be if this sum would maximally be 10 to the 6th, right? And what you see is that as this C increases, that as this resource constraint is relaxed, the system actually moves slowly towards this information bound, right? And that is, right, that is really because, right, is that this gain, right, so what really needs to happen is that the signal has to be lifted above the noise, right? And so you need gain for that. You need to amplify that. Now, the two ways by which you can do that, one is to increase this methylation time, but the other is to just have more copies of these proteins, right? So you can show that this gain depends not only on the methylation time, but also on the number of receptors and readout molecules, right? And so when this number is small, this gain that you need has to come from this methylation time, right? So the cell has no choice but to increase the methylation time, but this is basically then going to stretch the range of this exponential. You basically move this lobe like this, right? So this decays much more slowly now, and this means that the derivative now becomes a derivative taken over a longer time scale back into the past, right? So it moves away from this instantaneous derivative, which is the derivative that contains the most information about the future. But by increasing this range, it can more reliably determine the derivative, right? Because the gain is larger, so the signal is lifted more above the noise. So the derivative, while less recent, if you wish, does become more reliable. And it's this trade-off which sets this optimal methylation time. But if you can just simply increase the number of receptors and readout molecules, if that constraint is smaller, then you can get the gain from that, and then the system can afford to have a lower methylation time. Yeah.
So this question is related. So the resource constraint that is graphed, of course, is the path information. But I wanted to ask if there is a way to rewrite the resource constraint so that it's actually which resource is constrained, and such that the curve of maximum information coincides with the information bound. So like rethink the information bound in terms of the real resource constraints that are acting as which is the information that is being optimized, right?
Yeah, well, that is basically right. I mean, you can express this predictive information as a function of past information, right? That gives you that black curve, right? That is the information bound, right? And then you can also express this as a function of this resource cost, right? And then then you get basically. Right, then you get basically this region. This is the accessible region subject to a resource cost, right? And and then right. So then you see that these resources, they sort of limit this space that is accessible. So I don't know what you would precisely accomplish with what you have in mind.
Okay. So you could rewrite the resource cost in the like quantifying
right?
C, right? You could write predictive information as a function of C.
Yeah, you can write the predictive information as a function of C. You can write the past information as a function of C, and this graph then plots the possible combinations of predictive and past information for a given resource cost. And that's this area highlighted in green for a C that is at most 10 to the 4, right? For C at most 10 to the 2, it would be the area under div, and for C at most 10 to the 6, it would be the whole area under this dashed line.
Yes.
Yeah.
All right, interesting talk. the question is about, this is more of a conceptual question. If a system learns about how to behave in the future by learning about its past, its future is completely constrained by its past. Then where is the space for adaptation, phenotypic adaptation that happens within the lifetime? Meaning that there has to be some uncertainty for the system to adapt to novel situations. And that automatically should reduce the information bound the system has. Because if it is maximally, if it accrued maximal information, then there is no space for adaptation. That's what I'm trying to understand.
No, so, right, so what we assume here, of course, right, is that the statistics of the signal are given, right? So what we did here, right, in this calculation, right, is basically this applies to the regime, right, where the signal statistics is dictated by how the E. coli cell swims, right? And this gives rise to the signal statistics characterized by this two-point correlation function, right? And so this is the strategy that maximizes this predictive information for this signal statistics. Now, this is really determined by its own run and tumble dynamics to a large degree, and certainly at shallow gradients, right? This is, to a good approximation, the signal statistics it will see. But at steeper gradients or concentrations that fluctuate much more wildly, one would have to redo this whole calculation, because then the optimal design, indeed, in general, the optimal design always depends on the statistics of the signal, right? And what's nice about this calculation is actually, right, that it really applies to the regime of shallow gradients, because then we know that to a very good approximation, the temporal structure of the signal is really set by its own swimming behavior, which is sort of hardwired in the bacterium, right? And so then this really applies. But indeed, if the concentration fluctuates much more broadly, then one would have to redo this analysis.
Hi, I had a question on the costs.
Yeah.
So you're weighting receptor and readout molecules equally in cost, and that seems reasonable, but not inevitable. Would things shift around a lot if they had different weights?
Yeah, a little bit, but not much. So we have looked at that indeed, and so right. So you can ask, for example, right, if the cost of making the readout molecules were cheaper, right, then the system will do a little bit more of time averaging, right, because then it pays off to have more readout molecules per receptor, right. So it does more time averaging. So we see these things, but the effect is relatively minor.
Could you please show the slide where you have written down the expression connecting output signal to input signal with some kernel?
You mean in the beginning, right? This one.
Yeah, there is some noise term. Yeah, this one. I assume this is a white noise. Could you please explain what does this noise model and how the results depends on the characteristics of this noise?
Well, so you can take a network, you can linearize the network, you can look at the output, then this noise, right, that takes into account the noise that comes from receptor ligand binding and unbinding, that's colored noise. But it also includes the noise that comes from the signaling network downstream of the receptor. And so this is all lumped into this Xi.
So you assume...
So this is not necessarily Gaussian white noise, it could be colored.
But its time scale is much smaller than everything else?
So indeed, so typically, right, so the integration time, tau r, is about a factor of 10 larger than the correlation time that comes from receptor ligand and unbinding. And the methylation time is even much longer than that. That's another factor of 10, roughly.
So if that noise, correlation time of the noise becomes larger, how the results change? Do you have any idea?
So yeah, so right, so you can, so if the correlation time of the input noise becomes larger because receptor ligand binding, for example, becomes slower, concentration becomes slower, right? Then time averaging becomes harder. You need a longer integration time to have the same number of effective measurements, right? Because the number of effective measurements to a good approximation is given by the integration time over the correlation time. So if you make the correlation time of the noise longer, you also have to make the integration time longer. And of course, this is bad, right? Because then you at some point also start to integrate out the signal itself, right? And so what you will find is that then the information goes down and the optimal integration time moves up. Yeah.
Thank you.
Yeah, are there experiments or even theoretical results with two signals, like two concentrations or one concentration and temperature, for instance? Is the cell able to learn correlations and use it for predictions?
Excellent question, and the answer is no, unfortunately. No, it's beautiful. No, so this is something that we want to theoretically explore, indeed. So if you have two signals that are not only autocorrelated, but cross-correlated, what's the optimal system to then make prediction? Yeah, so that's an excellent question. Yeah.
Yeah, I would like to ask a question. As in your slide, your model of output signal is based on a linear integral equation. How would the theory could be extended, and what would be outcome if we model output signal by a nonlinear integral operator?
Well, so first of all, it would be much harder to make analytical progress as we can do here. That's the first comment. Second is that we actually know from experiments that systems like this are remarkably well described in the linear regime. So we have, this is known from other people's work, also from earlier work done in our group, but also more recently we revisited this problem. So certainly for the E. coli chemotaxis system in this regime of shallow gradients that we discussed, this Gaussian framework works extremely well. We compared that by comparing the results against exact stochastic simulations. But of course, if the signal in the environment fluctuates much more strongly, right, then at some point this will break down, right, and then the optimal system probably will be also much more nonlinear. But then you have to go to gradients that are much steeper, or signals that fluctuate because of other reasons. For example, cells that eat each other's signal, right? And then you can get all kinds of dynamics of the signal. But we have not explored that yet. Yeah.
To add to here, and what if, so one of the equations which we're using it was also concentration, which was also modeled by a linear equation. What if, let's say, the signal which a biological organism tries to predict is also governed by nonlinearities? Would it affect
If the signal itself would be very different. Right, so that is, I think, work that needs to be done. Right, so so here we took, right, so for the last part, right, we took basically as the signal, right, so we took here the signal that was measured, right, so this signal, that's the signal that actually was measured experimentally in this paper, right, and so well, actually this corresponds to the swimming behavior that was measured, and in the shallow gradient, this swimming behavior completely dictates the statistics of the signal, right? So this really corresponds to the experimental scenario in the regime of shallow gradients. But indeed, as I mentioned, right, when the signal starts to fluctuate much more rapidly, also because, for example, bacteria start consuming each other's food, right, then indeed this analysis would have to be redone. Yeah. Yeah.
Okay, I mean, we have another minute or two. They don't want us up to lunch till 12:45, not a minute earlier. So, and it only takes a minute to get upstairs.
So, you've considered this question with this specific signalling pathway.
Yeah.
And I just wonder, I mean, are there other pathways that might have done better?
Well,
is
so
this,
that's an
is this
excellent question.
phosphorylation, dephosphorylation? I mean, is that somehow an optimal signalling strategy, or would
Yes.
there be other ways of doing it?
Yes. Thanks for asking this question, because this is a message that was hidden, implicit in my talk. So now I get the chance to say it explicitly. So for this Ornstein-Uhlenbeck signal, this Markovian signal that I started with, this push-pull network is sufficient, right? It can come to the bound, right? And I don't think there's another system that would do better. For this derivative taking scenario, the E. coli chemotaxis system, that also can reach this information bound. Now, of course, this depends on the statistics of the signal, right? I also alluded to that in the response to an earlier question, right? And so we have actually started looking at more complex signals as given, for example, as described by a generalized Langevin equation, right? And so then you can get signals with a more complex temporal structure, right? And so we have analyzed what the information bound for that is, that we have worked out. You get more complicated kernels, that's also clear. But what the signaling networks are that can implement the kernels, and that would address your question, that we still have not figured out yet. But it's an excellent question.
Very nice talk. So this may not be the most relevant given the kind of things you talked about, but one can imagine this prediction paradigm coming in play for things like circadian oscillations when you sort of want to figure out day-night. So if you have time-varying signals of different sorts, do these kinds of ideas work?
Well, so we've done a lot of work in the past on circadian rhythms, right? And a circadian rhythm is really different, right? So because with a circadian rhythm, you basically have to know the current time of the day, and then you know that sort of time will evolve linearly, right? So if you now know that it's, let's say, well, what is it? It's almost lunchtime, right? Then we know that in a minute from now, we will have lunch, right? And so it's really like a sensing problem. If I know the time now, I know that in a minute I will have lunch. Whereas here, the future signal is inherently much more unpredictable.
So I'm thinking of circadian, like let's say we have a cloudy day.
Yeah.
I'm thinking of situations like that, where I have imperfect signals coming in. Can I apply these kinds of ideas to sort of think of?
Yeah, excellent question. So we thought about that. So the way we think about the clouds, etc., right, is that is something that you want to filter out to more accurately determine what the current time actually is, and so that's still different from this. Yeah.
All right, great, thank you.